Welcome to today's lecture on the fascinating world of microbiology, specifically focusing on the intricate details of viral entry mechanisms, genomic variation, replication strategies, and other key aspects of viral biology. Viruses, despite being simple in structure, possess an incredible ability to exploit host cells and cause a wide array of diseases. Understanding the complex processes by which viruses invade, replicate, and spread is crucial in developing effective antiviral strategies. So, let's dive into the 12 topics we'll be exploring today. Viruses have evolved various strategies to enter host cells, either by receptor-mediated endocytosis or membrane fusion. In receptor-mediated endocytosis, viral particles bind to specific cell surface receptors, triggering receptor-mediated internalization. This process allows viruses, such as influenza and HIV, to enter the host cell by forming endosomes. On the other hand, membrane fusion involves the direct fusion of viral and cellular membranes. This mechanism is employed by enveloped viruses, including HIV, herpes viruses, and the paramyxovirus family. Viral surface proteins, such as fusion proteins, facilitate the fusion of viral and cellular membranes. Some viruses, like retroviruses, can convert their RNA genomes into DNA through the process of reverse transcription. This DNA is then integrated into the host genome, allowing the virus to persistently infect the host. The integrated viral DNA, called a provirus, can be transcribed and translated by the host cells to make viral proteins. In summary, viral genomic variation includes antigenic drift, antigenic shift, and quasi-species. These mechanisms contribute to the evolution of viruses, their ability to evade the immune system, and the occurrence of influenza epidemics and pandemics. Viral replication can occur through two distinct strategies: lytic and lysogenic life cycles. The lytic cycle involves viral replication and cell destruction, while the lysogenic cycle involves integration of viral genes into the host DNA, allowing the virus to persist in a dormant state. In the assembly of enveloped viruses, viral budding occurs as viral proteins accumulate at the host cell membrane. This interaction with host cell machinery leads to the curvature of the membrane, forming a budding virion. As the process continues, the viral envelope incorporates viral genetic material, allowing the virus to exit the host cell with a lipid envelope derived from the host cell membrane. This is how viruses like HIV and herpes viruses acquire their outer envelope. During viral maturation, the newly formed virions undergo proteolytic cleavage of viral proteins, proper folding of viral proteins, and accurate packaging of viral genomic material into the viral capsid. This process ensures the production of infectious viral particles. In the viral entry complexes section, we examine the specific interactions between viral proteins and host factors that allow viruses to enter host cells. HIV, for example, utilizes the CD4 receptor and co-receptors CCR5 or CXCR4 for binding and entry. This binding triggers conformational changes in viral envelope proteins, leading to fusion of viral and cellular membranes. These interactions also determine the tropism of a virus, or the range of hosts it can infect. Understanding these mechanisms is crucial for developing effective antiviral therapies and preventing viral infections. Viruses exhibit varied host ranges, often limited to specific species or cell types. The ability of a virus to infect a particular host or cell depends on interactions between viral proteins and host factors, including receptors and co-receptors. Determinants of host range and cell tropism can be identified through studies involving viral mutants, receptor identification, and studies assessing viral infectivity in different organisms. These determinants play a crucial role in understanding viral emergence, zoonotic transmission, and cross-species infections. Additionally, host range determinants impact the development of vaccines and antiviral therapies by providing valuable insights into the vulnerabilities of specific viral strains. The viral capsid is a protein shell that encloses the viral genome, protecting it from external factors. Capsid assembly involves the self-assembly of viral proteins into a defined geometric structure. The structural organization of capsids can vary, with some viruses forming icosahedral capsids, such as poliovirus, while others having helical structures, such as tobacco mosaic virus. Capsid assembly occurs through specific protein-protein interactions and, in some cases, with the assistance of host chaperones. Understanding these self-assembly pathways is essential for deciphering viral morphogenesis and identifying potential targets for antiviral interventions. Viral genome packaging, which involves incorporating viral DNA into a protein shell, is regulated by specific nucleotide sequences called packaging signals. Different viruses employ various mechanisms for packaging their genomes, aiding in viral replication and evolution. Viral restriction factors are host proteins that prevent viral replication. They target different parts of the viral life cycle, like entry and replication. Viruses have evolved strategies to evade these factors, so studying this interaction could improve antiviral therapies. Therefore, understanding these viral exit strategies is crucial for developing effective antiviral therapies and preventing the spread of infectious diseases. In conclusion, this lecture discussed vital aspects of viral structure and replication, highlighting their importance in developing antiviral strategies, predicting viral outbreaks, managing viral persistence, and understanding viral host interactions. These insights contribute to advancements in diagnostics, therapeutics, and public health strategies.